My name is Rebecca Salamon, and I'm a third year here at UW in the lab of Dr. Ahmed Mahmoud. I chose to come to UW because I came from a small liberal arts college. I wanted to maintain that personal interactions I had with my professors and my peers, but get better access to technology. And I was really able to find that here at UW. The lab I work in is very small. There's only 2 graduate students and 2 postdocs, but I love that because I get a lot of one-on-one mentorship. My project focuses on looking at how the heart changes during development, disease, and regeneration. In order to look into this, we use the mouse as a model for mammalian heart development and changes during regeneration and disease. The mouse is a perfect model for this. First of all, it's a mammal like us, so it's very reflective of what the human heart does. Second of all, it has a very unique phenotype. Right after birth, the neonatal heart can regenerate. So if it gets injured, it can completely repair the heart, and there's no signs of injury. But just 1 week after birth, that flip switches, and you will see the heart will respond to injury with disease and damage, similar to what we see in the adult human heart. So my project focuses on understanding what occurs during that switch. So clearing the heart has really become a staple of my thesis project. I'm very interested in the cardiac nerves, but not in the aspect that you might have heard of most of them. A lot of times we talk about nerves in terms of changing the heart's rate, but my project really focuses on what patterns are allowing it to function in that way. There's a huge gap in knowledge and understanding the patterns of the cardiac nerves. You'd think we'd know that by now, but sure enough, we don't. And that has led further gaps in knowledge of understanding how those nerve patternings change after an injury or during regeneration. So in order to study that, I'll take an adult mouse heart. Actually, you can see that this one I've injured because you can see that little blue suture that I've put on the heart. That's how we injure the heart. And I'll send it through the clarity method. That's where we draw out the pigment from the heart. And I'll end up getting a heart that looks something like this. And the best part of UW is access to technology. Just 2 floors below me is an imaging core where I can get access to a 2-photon microscope to image this in 3D. Come on, I'll show you. So this is the imaging core. I work in the Wisconsin Institute for Medical Research, also known as WIMR. Here, we've got the 2-photon microscope behind me, and when the person who runs this learned that I was trying to do 3D imaging, they actually made me a 3D custom well, so that way I could put my sample in and image it on the 2-photon. Because of that, I've gotten some great insights to the cardiac nerves. This is one of my favorite images that I've gotten. And you can see, the heart's covered in nerves. None of these have been patterned before. So that gives me a lot of work ahead of me. But I hope you'll consider UW for your graduate studies. I've surely loved it here. I almost forgot to mention, I've also got a pretty good view from my desk.